Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will talk about detection and comparison of protein binding sites. I will explain this video in a scenario like let's assume you have a protein structure and you want to use it as a receptor. Last time we have covered a video about CLIPS database, but that was particularly for the kinase receptors. Although kinase receptors are pretty much higher in numbers, but there is a possibility that you may not deal with a kinase. So today in this video tutorial I will talk about how to do or how to compare the clips or how to calculate the binding site for your protein if you don't know before docking, and how to validate those binding sites with some statistical and some computational programs. Before going to start our actual tutorial, I also want to thank those people who sent me emails in personal on my personal email, and they actually encouraged me and they liked my work. I will not name them, but I received an email from Canada and also from few other countries like Latin America, Brazil. So thank you so much for your support, and I will try my best to educate all the youngsters or the new guys coming into the field. But remember, knowledge is just like an ocean. So my work is just a smaller drop there, and for most of the thing I'm also taking these information from other places and then combine them into some sort of story. So I will not claim that the whole work is I'm doing it. For most of my notebook I properly cite those guys who are the pioneer of this work. So thank you once again, and let's start. So we will work in the same Google Colab because it is easy for most of you guys. Okay, I will also drop this link in description section. Here you have to select the cat project, then you have to come and select detect binding site part 9. Click here. Now click here to connect. Okay, so this is about the introduction and those labs. You can go there, you can read their papers. Okay, what is the basic aim of this tutorial? As I mentioned in the start, that today we will learn how we can calculate binding sites in our protein. We will not only learn how to calculate, but we will also learn how to compare the results of two different programs, and then how to decide the binding site residues in our protein. Okay, last time in part 8, I guess, yes, part 8, we have covered clips, so I will not talk about in detail in about CLEPS, but today we will cover dog side scorer or DOG side scorer. DOG stands for difference of Gaussian side scorer, and we will cover this today. And finally we will also detect binding site by this method. We will then use the same method quickly, and then we will compare the result of this and the result of this at the end, and we will decide what are those residues which are particularly of prime importance. So this will be in our today tutorial. These are some references. If you want to read, you can read. Okay, what are protein binding site? I am sure most of you are well aware of the protein binding site, but let me in a single sentence just comprehend it, that it is a site where a ligand molecule or an organic molecule, or I would say some lead molecule, or in your terms you can say a drug molecule, is happy to interact, is happy to bind there, and the protein is also prone for that organic molecule in that specific site. So we call it a binding site or active site, whatever you call it. Okay, you need to understand few things. One is geometric based and the other is energy based. What it means: there are different programs in the field of computational biology. Some of them decides, or I would say some of the algorithms of these programs are deciding based on the geometry. If I comment how they decide based on the geometry, so normally I give you a very straightforward example, because most of you are well aware of key and lock model. Just like if you suppose that protein is a lock, and there for every lock there must be some specific key which fits into that specific pocket of that lock. So if you think on this example, or if you think on this theory or law, the key and lock model, so it means you are thinking based on geometry, because the structure is related to the geometry. So there are some programs which decides the binding sites or the residues or amino acid present in the binding site based on the geometry. So then there are some algorithms which decides the binding site based on the energy. How they decide based on the energy? Remember, rule of thumb. I call it rule of thumb. If you are working in a protein science, or if you are working with a protein, the minimum energy means the native protein. And I don't want to start another debate, but this is the one reason that the higher negative score you achieve from any docking program, we call it the better score, because the minimum energy is actually the best fit, because in scientific term it describes the native state of the protein. Also, I hinted in my last video that the minimum energy theoretically it should be between -20 and -60. It's a rough calculation. It doesn't mean that -15 will not be a good choice, but This is from experiments and from scientific calculations. These are some established values, minus 20 and minus 60 for the protein, actually, not for the single or two or peptides, for the whole protein. It's okay. So there are two types of algorithms. One decide based on energy and the other decide based on geometry. Now both of these algorithms, they decide in two different ways, either grid based or grid free based. In grid based methods, what is the difference actually? So if you are familiar with the docking, in docking we are using grid maps. What are actually grid maps? If you know that, you will understand here. Grid maps are actually the coordinates position, I would say, of that particular atom you are dealing in your compound. If the compound is a protein or an organic molecule, that doesn't matter, but I use the word atom. So the position of a particular atom in your sample, I would say proteins or whatever you organic molecules, so the position of that particular atom on the map, or the location of that particular atom on the map, is a kind of grid map. So those algorithms which deals proteins using those coordinate information, they will be grid based, and those which do not deals the specific location or the orientation or the conformation of these molecules, then you can call it grid free methods. I also suggest you to read about grid based and grid free method if you are still confused. So there are some programs just like in geometric based, Lakeside and Surfnet. Surfnet is then grid free, geometric based, Lakeside is grid based. In energy method, Drugside is grid based and docking is grid free. Okay, now these are those explanations I provided here, and these are with the papers information. So if you want to read it, just copy paste here in your Google. Okay, now today we will talk about Dockside Scorer. Dockside Scorer is actually a program which incorporates geometric, and then in the category of geometric it is a grid based method, and it decide with the mathematical value like 0 or 1. If it is 0, the program understand that this region is free. If it is 1, the program understand that this region or this particular atom is not free for any other organic molecules to come and interact. So this way the program decides the pocket residues, and its name, the DOG or dog, as I mentioned earlier, it's from difference of Gaussian, and the dog site, it's also called the dog site, and the score it provides is called dog site score. It's pretty much powerful program, and it also calculates some druggability. What is druggability? So this program decide it based on some statistical calculation and some support vector machine, or we call it SVM model. How it calculates the druggability? So in simple terms, if I tell you that if a protein has the ability to bind some molecule at some specific site or some specific region, then the program can count, or the program can mark that region is more druggable as compared to the region which is not exposed, which is not free, which is occupied. Then in that case, that area will be less druggable as compared to the one which is exposed to the binding will be higher druggable. So these are the kind of predictions these programs are performing. So that's why, although I work in the field of bioinformatics and structural biology, but that's why I always tell to my students that this is a predictive science. Don't indulge in some discussions and don't say that this is 100% correct. Remember, if you are a scientific student or if you are a student of science, so there is always a room for a research. So nothing is perfect or nothing is 100% correct. There is a room for further research. So this is also the druggability. This is another debate, how much it is correct or how much it is wrong. It's not point of this discussion. Okay, so this is just a schematic representation. I discussed some of them, 0, 1, free or not, or based on energy. The lower energy, it means the bitter side, the higher energy, not the bitter side. Just like what are sub pockets, we will discuss it later on. But sub pockets means the part 2 of the main pocket. Let's say if you are living in a house and your house has one drawing room, but there is also a smaller corner or a smaller room which can also be used as a sub drawing room. So in that sense you can say that this house has a main drawing room and sub drawing room. The same way in protein, the one is the main pocket and others are the sub pockets. If there are some smaller holes which present some druggability, in that case we call it sub pockets. Okay, and I have covered this in part 8, so I will not cover it here further. Okay, let's come to the practical. First of all, we will do some installation. So let me start it and I will pause this session just not to waste your time. Okay, welcome back. It may took 2 to 3 minutes all the installation. So let's proceed. Now, binding site detection using dock site scorer. So first of all, we will generate some API token because the dock scorer is present on the server. Its name is Protein Plus and it is hosted by University of Hamburg. So to access this, first we will create some token. Remember, to create your token you have to mention your PDB ID here. Let me first clear here, if you want to use your own structure, then I have not tested it yet, but let me tell you in advance that in that case you just need to see my other notebooks, because even in docking notebooks in many places I have uploaded the structure from the local computer. So you can copy that code here and then do it that way. Let's try it. I have not tested it yet, so I cannot say whether it will work or not, but it needs or it is supposed to be working. So how you will incorporate a new code here, just say, you can see here code, you bring your mouse here, you can see code and text. So just code, and now copy that code from another notebook and paste it here and run it. So you will be able to upload your protein, but so far I'm not sure whether you will be able to work with it or not. So right now I should announce that this is supposed to be for the PDB protein. Also, if your protein is not present in the PDB, then don't worry from where you have modeled it. It's a question from my side. Possibly you have modeled it with using homology modeling, or let's say Swiss modeling server. In that case, the Swiss modeling server has used a homolog template. So you can use the PDB code of that homolog template also, because it's homolog to your protein. So normally the binding site residues are conserved, so you will have the same residues in that protein as well as in your protein. So I hope I have answered this question in advance. Okay, now we are going to generate some API, but for the API you need to edit this part with the actual PDB ID, and you need to edit this part, the ligand ID. Now how will you know your ligand ID? So actually in the ligand ID, it will be in the protein plus server. This will be the ligand information, this will be the chain information, and this will be the first three letter, the last three letter of the protein. So let's say this is our protein. I say I provide my protein 3W32, 3W32. So let's say 3W32, and let's say go. Oh, I mentioned 3W3S. It's my mistake. 3W32. Go, and you will have a ligand ID W32A 1101. Now this is the chain information also present here. I told you this is the protein and this will be the ligand. So you have to copy this whole name here. This will be your protein name, this is the chain. And now don't change some other thing, just run it. Once you run it, it will generate some token. Now here we will submit our job, but once again you need to edit this name, you need to edit the chain ID, and you need to mention the ligand ID as we mentioned above. Run it. The job has been submitted, and it is a kind of location, but it's not for us, it is actually for this notebook. So don't worry, you can clear it. Okay, now you have to run this step. In this step actually we are generating some data frames from the data which will be calculated, or which has to be calculated in the protein plus server. Now run this. This is the data we got it from the protein plus server. You will get the data for your protein. Okay, just understand few things in this data. So the name, if you see P_0, so if you see P_0 is common in all, it means in this protein there is only one main pocket, and there are also some sub pockets. So if you see P_0 for your protein, and then you see P_1, and then you see P_2, it means these three main pockets. But if you see p underscore 0, then 0 1 0 1 2 3, but this area remains the same, it means it's only one major pocket and these four are the sub pockets. Another thing, you have to check the ligand coverage and you have to check the pocket coverage. The main site, or the main, I would say the whole site, or the whole pocket, its ligand coverage is this, and its pocket coverage is this one. Both these values, if they are higher, they are better. If they are smaller, I would not say that they are not better, but it means the pocket is smaller. But in docking, we want a slightly bigger pocket so that they can accommodate different kinds of ligands. Simple. Okay. Now p underscore 0, this is the subsite. So p underscore 0 0. So if you can see, the pocket coverage of this subsite is greater than this one, and also you can see some druggability information. Okay, probably not here. Okay, come down. Now what we did, we just choose all these names, all these columns, and we ask the program just to remember it. Why? We will see later on. Okay, now we ask the program to make this data in descending order, the higher in top and the smaller at the bottom, but based on these two columns, ligand coverage and pocket coverage. So now you can see the sub pocket p dot 0 underscore 1 is on the top, because the pocket coverage is also higher here as compared to this main pocket. Okay, and let's see, there is also some drug score. Now you can argue that the drug score of the main pocket is higher than this one. So this is your choice, whether to choose this pocket to analyze this pocket, or to analyze this pocket, or to analyze both. So it's up to you, but this is almost 62% druggable, the subsite, but the main site is 81% druggable. Okay, now we will make some other functions. Just click here, and what these functions will do, we will see. Okay, based on the ligand coverage and based on the pocket coverage, we have selected POO, or p underscore 0 underscore 0, which is a sub pocket for our analysis. Okay, you can also choose another one and another pocket, but how? Can you see here, get name of the pocket? So I use the index number 0, because the top one, it will be called 0, then 1 2 3. So if you choose the index number 1, you will be able to select this pocket, but here we are going to select this the sub pocket here. Okay, so this will be our pocket for analysis. Now we are asking, we will take the binding site information of this pocket. So we will run this. We will also run this, and now we will run this. We will have some kind of URL, but it's not for you, it's actually for our notebook. Now this is the information for our sub pocket p underscore 0 underscore 0. These are the titles. We asked the notebook just about to remember it. Now we ask the notebook that complete this title only for this pocket. So you can see all the information here. Okay, now this is about the visualization, but today I tried my best to fix this visualization. But remember, in visualization we are using two different things: NGL view and widgets. But sometimes they are third party software, sometimes they are not happy with notebook and you will have some problem. But don't worry, in today's tutorial, if I have placed all the codes, I hope in your computer you will be able to visualize these pockets. But if you are unable, so don't worry, it will not affect anything here. I will show you how. Okay, just click here. Now there is some more installation, but it will be for less than a minute. Okay, done. I have installed BioPython. Now I am calling the NGL view again. I'm asking the Jupyter notebook to, you can see it's not recognizing the NGL view, and that's why we will not be able. But it might be you in your computer maybe it worked. But if you are failing, so that's not a major problem. It's okay, ignore it, just clear. Okay, now click here, and to see whether you are able to view some data, just cross here, whether you are able to view some data or not. So as I told you, I'm not able to view some data, but that's okay. If you can view, so that's okay. You can also take pictures running these two, otherwise leave it. Now what we did, we just detected the dark side scorer. We just run we just use our protein, and then we detected some pockets, and then we have selected the first pocket, not the main pocket, and we selected this pocket based on the ligand coverage and based on the pocket coverage. Now quickly we will also compare this pocket based on the clips, and then we will compare the result of dark side score and clips, and let's see what happens. The same thing, we will generate some token. Now these are the residues we got from the dark side scorer. But remember, these are not only 5 residues. We have just used the hit function, so we just printed 5. There are many. You can see the index number. This is the 20th. So let it be. Later on we will call all the residues. Okay, again the visualization script will not work, but let me run it. But that doesn't matter. if it is not working, it will not affect anything. It's just a repetition of the same script, because I was working with this notebook, so I forgot to delete this. But that's okay. If it's not working, so it's not a major worry for us. Okay, so now get clips pocket residues. We have covered this section in part 8, so here we will just cover it quickly by running the main scripts. So we asked the program like to use and got the structure, the PDB ID. The PDB ID we have mentioned for the program in the start, 3W, 3S or 32, and then we asked like to detect the pocket residue for 0. What is 0? 0 is the index number of the first sub pocket. So we did it. I will clear it, doesn't matter. Now we are calculating clips, and once again this is the hit function, so it doesn't mean that the clips only calculate 1 2 3 4 5 residues. No, the clips calculates so many. Okay, now get dog site scorer pocket residue as a list and get clip pocket residue as a list. If you read it, for this pocket or for this sub pocket, 29 residues were detected by the dog site scorer, and clips detected this much. That's interesting. Clips detected more residues as compared to the dog site scorer. Now we will do some overlap analysis. Okay, 27 residues among 29 which was detected by the dog site scorer are common. Now just imagine that you are working with clips, only with clips, not with other stuffs, not with other algorithms. So you will have this huge amount of residues, and it's so many. There are so many, and even a child also knows that in a protein 85 residues, they are not important as in a pocket. So what happened with this comparison, that now we are sure, we have some scientific arguments that the dog site scorer calculated 29 and the clips calculated 58, then 85, then 27 were common or 27 were overlapping. Okay, this is the complete list. That's why I told you that if visualization is not working, that doesn't matter. You can visualize these residue of your protein in PyMol or in any visualizer you are working. So these are the numbers. The first line represents the residues detected by the dog site pocket residues. These are the dog site pocket residues, and the second lines is the clips pocket residues. So the clips pocket residues are more, as we are aware that why they are more, because here we have read it, and the dog's pocket residue will be here. If you are confused that why there are numbers, not names like histidine, alanine, valine, so these are actually the index number of the residues. When you open your structure in any visualizer, so you just need to click the 718, so it will pick that residues which is present. Now we will, so from here you can also find out the common residues, or diagrammatically you can find it here. It's the dog site scorer. It's I would say this ball is for the dog site. Now if you can see, only 2 residues which are predicted by the dog site, and they are not present with clips. The 27 residues are in common. It means that these 27 residues are of more importance as compared to the other overall residues. Okay, now once again, if you want to find out these analysis for another pocket, let's say p dot 0 or the main pocket, so you don't need to repeat the same stuff. Just write here, let's say p underscore dot 0, this is the main pocket, so I just clicked here and the dog side calculated 62. Here it has calculated 29. Okay, now how about the clips for this one? Yes, the clips are almost, I don't know how many of them, but only 45 amino acid are in common. If you want to calculate for some another, then you can. You just change this name p dot 0 dot underscore p underscore 0 underscore 1, then also here just change this name to p1. Or if you don't know how to do it, so you can repeat the whole from here, you can repeat from here and just ask the program to select another, to select this one or this one, whatever you like, and then calculate. Okay, how you will get the information about those residues? So it's from here. Okay, how this tutorial will help you? This tutorial will help you in many ways. If you are publishing your results, so I'm always against the blind docking, so you need to select a region with some scientific way. So this is a kind of scientific way that you have performed cliff and you have performed dockside scorer, and then the combined residues fraction like 25, 27, 13, whatever they are, we have used those residues in our grid box and grid in those settings, and we generated our docking this way. So your docking will be more powerful and more scientific. I hope I have covered many things in this tutorial. So still, if you have any question, you can drop your question in comments, and most of you sometimes they ask questions which are already covered in the video, so normally I don't answer them. So the same way I will not answer those questions which are covered in this video. So if you have not subscribed this channel, please subscribe and share this channel with your friends and colleagues. This is a request or suggestion, and if you are liking my videos, just provide thumbs up. Thank you so much. Bye bye.